Hey, food science. We are going to be talking about kind of osmosis a little bit more today, but we're going to be talking about it in the context of our food science class. So osmosis is something that's really important that happens in your body to help your body function properly, but it's also a technique that they use in a couple different things for food, and I think it's kind of cool to talk about. So we're going to get into that a little bit today. Just as a start to review, I'm going to give you a couple terms because I think that the video that we previously watched is good, but it's going to help to have a couple things defined a little bit. So the first one we want to talk about is solution. Now, when we talk about the term solution, what it means is it's a homogeneous mixture of a solute and a solvent. And homogeneous means it's the same throughout. So like nothing settled on the bottom of the container when you look at it or anything like that. And the two terms that are really going to be important here are solute and solvent. So the solute is the substance that's dissolved in a solution. So if you think about making a Kool-Aid solution or, pitcher of Kool-Aid, the solute in the Kool-Aid solution is going to be the Kool-Aid powder and the sugar. You'd have two solutes in there. So the stuff that's being dissolved. And then the solvent is the thing that you're dissolving the solute in. So most of the time, especially in food science, your solvent is going to be water. Not always, but most of the time. And when we talk about osmosis, our solvent is always going to be water. So the thing is, the amount of solute and solvent is really important in predicting what is going to happen with osmosis. In osmosis, what ends up happening is that you have what we call a selectively permeable membrane. And that membrane has tiny holes that let certain particles pass through it, but not other ones. And more often than not, they are big molecules that can't get through the membrane, and then little guys can. And they can change the sizes of the holes of those membranes to let different size molecules in and out. And so what drives the movement of the water, if I have, let's say, two solutions, here's my membrane, and I have two solutions on either side of that, and one has a different solute concentration compared to the other, the difference in concentration is going to force water to move across that membrane until we get our two concentrations equal on both sides of the membrane. Okay? The key to remember about it is that water is going to flow from the place with less solute to more solute. Or I think about it sometimes as it moves from the place where there's more water to less water. And you will get movement on both sides of the membrane. So a lot of water will head one direction, and maybe just a little bit of water will head in the other direction initially. And then as the reaction gets closer to, or not the reaction, the two different sides of the osmosis process, as they get closer in concentration, the amount of water that starts to pass back and forth between that membrane is equal. Okay, so equilibrium is what we call it, and it's reached when enough water has moved from one side to the other so that we have equal concentrations on both sides of that membrane. And at that point, when we hit that, the flow of water back and forth should be equal, like I said. So here is an example. We have side A and side B. If you take a look at side A, we're going to assume that these little circles here are the solute particle and the empty space is water, okay? And then we've got that selectively permeable membrane. And a membrane, like I said, it's just like a little membrane with holes in it, basically like a barrier with holes in it. So when I look at this particular solution and I want to figure out which direction the water is going to move, I have to look at kind of the density or concentration of how much solute is in each side. And if you look at side A here, I have a lot of water and not a lot of solute. And on side B, I have much more solute and therefore less water. And so what's going to happen is water from side A is going to move into side B a lot. So you're going to get a bunch of water moving to side B. Maybe a little bit from side B comes back, but the majority of the water is going to push over to side B. And what's going to happen is if I take water out of side A, the concentration over here on side A for my solute is actually going to go up. Because if I have the same amount of particles, but now they're dissolved in less water. That's going to be a higher concentration. On side B here, I'm going to have a concentration that's going down because I have the number of particles that I have, but now I add more water to it. And so what will happen is the water will move from side A to side B until we get those two concentrations equal. Okay, so A is going to get more concentrated, B will become less concentrated, and then they'll equal out and reach that equilibrium. Now, we talked about it, and they talked about it a little bit in the video that you watched the other day about your blood vessels. And water is really important when we are dealing with our bodies, which we've talked about a lot in this class. But when you're dealing with your blood in particular, it's really, really important that you don't actually get too much water in your body. It is possible to actually drink too much water. And if you drink too much water, what ends up happening is that if this center blood cell here, red blood cell, is our normal solution, what happens is we have a concentration of solute in the cell that's basically the same as the concentration of the solute outside the cell. If you don't drink enough water, the concentration of the stuff around the cell, the solute, is going to go up, and then it's going to pull water from that cell, and it's going to make you dehydrated. But on the other side of things, if you drink too much water, it dilutes or brings the concentration down for the solution that's outside your red blood cell. So what happens now is I've got a lower concentration of water here and a higher concentration of solute in here, so less water in the cell. And what'll happen is the water goes into your cell, and they do not have the structure to stop that from expanding. So what happens is eventually, if you get too much water, it can break your blood cells, like literally pop them like a water balloon that's too full. And if that happens, it can cause cellular damage and death. It's happened to some people who did like a water challenge where they tried to drink a ton of water in a short period of time, and then they died from it. So you have to be very careful that you don't drink too much water. It's a lot of water that you got to drink, but you do have to be careful. Okay, now it's food science, so we're going to talk about food, right? There are different food applications where they're going to use osmosis, and one of them is brining, which we'll talk about in a second, pickling, which we'll also talk about. They can actually use a process called reverse osmosis, where you pull the water out to make fruit juice concentrates and make whey protein. Like if you guys drink protein shakes at all and you add whey protein to it, they use reverse osmosis to actually pull them and make the solutions more concentrated, and also reducing waste streams. So these first two are normal osmosis. These second three here are reverse osmosis, where they try to pull the water out. We're going to talk about brining and pickling. So when we talk about brining is a situation where they take a piece of meat and then you put it into a solution that's got some salt water and stuff like that. So here's what I want you to take a look at. Imagine that this is your semi-permeable membrane. That's the layer of kind of skin between the meat and the solution you're going to put it in. And this is our meat. So all these particles here represent different kinds of solutes that are in there. And then this represents the brine. What do you think is going to happen when I put my piece of meat into the brine? So think about which way the water is going to move. Is the water going to move from the brine into the meat, or from the meat into the brine? Stop and think about it for a sec. Okay, so because the concentration of solute is less here, it means there's more water on the brine side and less water on the meat side. So what's going to happen is the water is going to go into the meat here, and then in theory, it's going to make your meat a little bit juicier. Also, what happens is if you think about this as being like salt, salt will also diffuse slowly into the meat and provide seasoning as well. And so it's going to move from the brine side to the meat side, the water will, and go into your meat. I kind of like this picture. So if you think about this as your meat here, then the water is flowing in. The other thing that salt does, which is kind of cool, is it breaks down some of the tissue here on the very surface of your food. And when that goes in there, when it breaks that down, normally when you cook meat, the proteins that are in there shrink up and it squeezes out a lot of the liquid. If they're broken, they don't squeeze out, and that also keeps your meat more tender as well. Okay. The other one is Pickles. Now, I guess I probably shouldn't have this being a pickle. It's a cucumber before we get started. But in a cucumber is like, I think like 98 or 99% water. So there's a little bit of solute that's in your cucumber. And then you have your brine. So if you think about this now, a pickle, when you start off as like a nice firm cucumber to begin with, and you put it into a brine that's got some vinegar, some salt, some dill, all that kind of stuff. But there's a lot of water in the cucumber. And then when I put it into the brine, the water travels from the cucumber into the brine, and then it pickles it. So it shrinks down, makes the water kind of go out of it, and then the flavoring actually diffuses into it. So those are just a couple applications that you can kind of see. And over time, when you pickle something, if you think about this as the beginning, like I said, lots of water. This is a lot of solute and a little bit of water. Over time, we reach that equilibrium where the concentrations of both things are equal, and then we end up with a pickle. So just a couple of different applications for osmosis to kind of give you a little bit of a deeper understanding. We're going to take a look at an activity next, so just be prepared for that. And if you have any questions, come to the meet and get things cleared up. Otherwise, good luck.